Moving on to some more bacterial diseases, and the next one is typhoid, or which is also called enteric fever. Now this disease is caused by Salmonella typhi, and this bacteria is spread through the fecal-oral route, which means that the infected person is passing out the bacteria in the feces, and these feces, if these are not properly disposed, will be contaminating the food and water of the other person. The symptoms include sustained high fever, which is of the order of 39 to 40 degrees Celsius, weakness, stomach pain, constipation, headache, loss of appetite, and in severe cases there might be intestinal perforations. And when there is intestinal perforation and intestinal content come out into the coelom, it infects the entire peritoneum, and this peritonitis proves to be fatal. Now this disease can be diagnosed by Vidal test. There are vaccines nowadays available against typhoid, and this vaccine is an oral vaccine also, having 4 doses, or it could be also a single dose injection. Now with typhoid there is one another problem, that is of relapses. Now typhoid bacteria, when it is in the intestine, it moves to the liver via hepatic portal system, and from the liver along with the bile juice it comes into the gallbladder. The gallbladder becomes the reservoir. So when we are taking antibiotics to cure ourselves from typhoid, the bacteria is removed from the intestine, but after the antibiotic dose is stopped, again bacteria can come from the gallbladder along with the bile juice and cause the infection once again. And sometimes the relapses are so common that the gallbladder needs to be removed for such patient. However, there are some people in which the bacteria remains in the body, but they don't have any symptoms, they don't have the disease. Now such people are called the human carriers. So the discussion of typhoid won't be over until unless we talk about Typhoid Mary. Now Typhoid Mary was cook. She was actually Mary Mellon, who was Irish immigrant to US, and she was working in various homes as a cook. Now in whichever home she was working, the people were getting typhoid, and since at that time such wide spectrum antibiotics were not available, so the disease was having a high fatal rate as well. So this female was then considered as a human carrier, and she was put in a quarantine hospital. Now we know that this bacteria can remain in the body as a carrier, so that is why we have to be careful who our food handlers are and how they are handling the food, whether they are maintaining proper hygiene before handling the food. So this also needs to be taken care of. Now moving on to the next disease, that is cholera. This one is caused by Vibrio cholerae. Now Vibrio is a type of bacteria which are in the shape of the punctuation mark, that is comma. Now the mode of transmission is fecal-oral route, and houseflies act as the mechanical carriers. The same thing, that is when the person who is infected with the cholera bacteria, they have diarrhea and vomiting, and if the feces and the vomitus are not disposed properly, they are in the open spaces and houseflies are sitting over there, then they would be transmitting the bacteria to the food and water of the other person. So in this case, there is copious watery diarrhea, which are usually called rice water stools, vomiting, and as a result there is loss of fluids from the body resulting in dehydration. And cholera these days is considered as a socio-economic indicator for a country, because in a developed nations where there is proper sanitation, there is no spread of cholera. But in a developing nation, the spread of cholera does occur because sanitation is not proper. So that's why it is a socio-economic indicator. Now moving on to the next bacterial disease, plague, which is also called Black Death. At one time, that was in 1346 to 1353. This was the reason behind the death of nearly 75 to 200 million people. Now, this disease is caused by a bacteria, Yersinia pestis, and this is not a human disease actually. It is a disease of the rats. The rats are infected with Yersinia pestis by the bite of a flea, which is an insect, Xenopsella cheopsis. Now, when the rats die, then the fleas, they leave the body of the rat and they infect the humans. So, the humans actually become the accidental host of this bacteria. Now, the symptoms here include high fever and buboes, which are lumps in the groin and the armpit, even in the neck, wherever there are lymph nodes. Those lymph nodes will undergo swelling and they would be protruding out, and these will then rupture, from which pus will be released. So, red patches appear all over the skin, which turn black and ultimately leads to the death. And because of this, it is also called the Black Death. Now, this disease, again, because of proper sanitation, has been restricted, it has been contained, although it is not as yet eradicated. So, as more and more crowd is filling up in these spaces, disposal of waste requires more space, and all those sewer rats are there, and those sewer rats, when they come in contact with the humans, there can still be the infection of plague. However, as we said, it is contained as yet. The diagnosis for plague can be done by Weissenstein stain test. Now, these were some of the important bacterial diseases, and from here we'll move ahead to the next disease, that is the protozoan diseases. Now, protozoa are unicellular eukaryotic organisms which are without a cell wall. And in this, one major disease that we have to discuss is malaria. Now, malaria, as the name denotes, it is mal plus air. So, earlier it was thought that this is a disease due to bad air, as it was found in regions nearing the swamps, and swamps are the breeding grounds of mosquitoes. So, it was much later that Dr. Ronald Ross, who was a British army doctor posted in Kolkata at that time, and he found that it is the mosquitoes, that is the female Anopheles, which is the vector of the disease, and it is Anopheles which spreads the disease. And for this finding, Dr. Ross got the 1902 Nobel Prize. So, malaria is caused by a protozoan by the name of Plasmodium, which, as we said, is transmitted by the mosquitoes. And in the human body, the parasites multiply in the liver and then they infect the RBCs, the red blood cells. And for this Plasmodium, there are nearly 60 different species known to us, but thankfully only 4 of them cause malaria. And those are Plasmodium vivax, ovale, falciparum and malariae. Now, out of these, the malaria caused by Plasmodium falciparum, that's the most dangerous one, and it is called the malignant malaria. And this malaria can be fatal. Now, the Plasmodium parasite spends its life cycle partly in the human host and partly in the mosquitoes. So, this way we can say that it is a digenetic parasite, and when a digenetic parasite we talk about, that means it requires 2 hosts to complete its life cycle. Out of that 2 hosts, one is called the primary host and the other is the secondary host. Now, the primary host here is humans. Now, normally in our earlier discussions with animal kingdom, we have mentioned that the host in which a parasite carries out its sexual cycle, that is considered to be the primary host. Now, for Plasmodium, this is slightly different because the sexual cycle begins in humans, but it is completed in the mosquito. But we will take some more criteria to consider humans as primary host. That is that out of vertebrates and invertebrates, if 2 hosts are there, then we have to consider vertebrates as primary host. and then warm-blooded and cold-blooded. So then two warm-blooded animals are to be considered as primary hosts. So these two criteria certainly are fulfilled by humans. Now this parasite shows alternation of generation as well as alternation of host. So alternation of generation is that there is haploid cycle as well as a diploid cycle, and alternation of host is of course that is humans and the mosquito. Now if we see the life cycle of the Plasmodium, it has number of stages. So if you see these stages over here, you will find that there are some stages which occur in the humans and some stages which occur in the mosquito. Now in humans, the stages that occur are the pre-erythrocytic schizogony, which occurs in the liver. Then there is erythrocytic schizogony, which occurs in the RBCs. Then there is gammogony, which begins in the RBCs, but it is completed in the mosquito's gut. And finally, there is sporogony, which occurs on the gut wall of the mosquito. So these are the various stages. And looking at this life cycle and the various stages, I'm very sure that you have found it very daunting. So not to fear, because each of this stage will take 2 bits and see what all is occurring in each of the stage one by one. Now taking up the life cycle of Plasmodium, so starting with the pre-erythrocytic schizogony. Now in this case, as we said, that the vector for malaria is the female Anopheles mosquito. Now the infected female mosquito, it bites the humans, and while it is taking the blood meal, it first of all releases its saliva. The saliva of the mosquito is containing an anticoagulant substance, that is, which will not allow the blood to clot, and during that time the mosquito can take in the blood. However, if this Anopheles is infected, the infective stage of this Plasmodium called sporozoites, this is present in the salivary gland. So along with the saliva, these sporozoites are also released into the human bloodstream. Now once these sporozoites, they enter into the human blood, they will quickly move to the liver. Now some of them will be killed in the path by the WBCs, but few sporozoites, if they are able to reach the liver cell, the cycle begins. And this first cycle is the pre-erythrocytic schizogony, which is occurring in the liver cells. Now this sporozoite which has entered into the liver cell, it will take up a spherical shape and it will undergo asexual mode of reproduction. And here the asexual mode of reproduction is multiple fission. And this term schizogony is actually for multiple fission only, where the parent nucleus divides into number of daughter nuclei, and then each daughter nucleus accumulates cytoplasm around it, and finally the daughter cells will be formed. Now the daughter cells which are going to be formed in this manner by asexual reproduction, those are going to be called merozoites. Now these merozoites, they are liberated from the liver cell. The liver cell ruptures and these merozoites are released. Now these liberated merozoites will re-enter further liver cells and repeat the same cycle. So this pre-erythrocytic schizogony is repeated 3 times, and as a result what happens is that the number of merozoites are tremendously increased. Now, after completing this cycle thrice, there would be two options with these merozoites. Some of them will enter the RBCs and start the next phase of the cycle, that is the erythrocytic schizogonia, while some of the merozoites will remain in the liver itself and continue to multiply in the same manner. However, now the cycle which will be in the liver, which is going on parallelly with that of RBCs, is going to be called exoerythrocytic schizogonia. And it is this cycle which is responsible for relapses. So even for malaria, there are number of relapses because our liver itself has become the reservoir for the disease. Now, out of the 4 various species of Plasmodium that we mentioned, out of that, Plasmodium falciparum lacks exoerythrocytic schizogonia. So there the merozoites which are liberated from the liver, all of them will be entering the RBCs. So there won't be any relapses. So moving on to the next phase of the life cycle, that is erythrocytic schizogonia. Now this one is known by other names also, like Golgi's cycle. So once these merozoites have been released from the liver, they invade the RBCs where they undergo another asexual cycle which is called erythrocytic schizogonia. Now here we can see various stages. Once they have entered the RBCs, they again take up a spherical shape and they feed on the contents within the RBC so that a large vacuole is formed in the center, which is called the signet ring stage. And this feeding stage is called the trophozoite. So the merozoite develop to form an immature or ring stage, which is the signet ring stage, trophozoites, which then progress to form the mature trophozoites. Now once this has occurred, it will take up amoeboid shape, and that's why it is called the amoeboid stage. And at this stage within the RBCs, there are some abnormal granules formed which are stained by eosine stain. So that's why these are called eosinophilic granules. And these granules have been named Schuffner's granules or Schuffner's dots. Now here, once it has reached the amoeboid stage, it will again start schizogonia, that is the multiple fission. And the daughter cells formed as a result of multiple fission, the terminology for it is going to be the same, that is merozoites. So merozoites are formed within the RBCs, and finally the RBCs are ruptured and these merozoites are liberated. And when these merozoites are liberated, so there is also release of hemozoin. Now hemozoin is a toxic substance which is formed by the breakdown of hemoglobin which was present inside the RBCs. Now in the hemoglobin, we know that iron is in the form of ferrous, but in hemozoin it is in the form of ferric. And it is this hemozoin, when it comes in contact with the tissues, there is the characteristic symptoms of malaria, including chills with high fever. So the person will feel rigors, the person would be shaking as if he is in a totally cold environment, and there would be high fever. And after this high fever, there is going to be sweating and the fever subsides. So this is a very typical symptom of malaria. So one thing we need to remember here, that the first symptom of malaria will be evident only after the rupture of the RBCs. And as far as Plasmodium vivax is concerned, for which the life cycle we are studying, this will occur after the 14th day. Now this time period, since the person is exposed to the infective agent, that is the sporozoite, and the first appearance of the symptom, that is called the incubation period. And here the incubation period is 14 days. Now the merozoites which have been released from the RBCs, they have 3 different paths that they can follow. One is they can invade RBCs and continue with the same cycle, and every time the cycle is completed and RBCs rupture, the same symptoms will appear, and that for vivax would be 48 hours. Then some of them can re-enter liver. They can return to the liver and enter the liver, and continue with the similar cycle as we had studied in the liver. However, now the name of the cycle that we use is post-erythrocytic schizogony. So you see, in liver there would be 3 levels of cycle. The first one that started with, that was pre-erythrocytic. The one which is going on parallel with the RBCs, that is exoerythrocytic, and one in which the merozoites from the RBCs have re-invaded the liver, that is the post-erythrocytic. The other merozoites, they will invade RBCs, but they will start with the sexual cycle, and that is called gamogony or gametogony. So taking up gamogony. Now gamogony, as we mentioned, that it begins in humans, but it is completed in the mosquitoes. So merozoites develop into gametocytes. So gametocytes are the precursor of gametes. So it is not yet gametes, but gametocytes in the RBCs, and it marks the beginning of the sexual cycle, which begins in humans, as we said, but is completed in the mosquito. Now when a female Anopheles mosquito bites a malarial patient, so it takes up the blood, and along with the blood, the different stages of the parasite will also go into the Anopheles, in which there would be both the male gametocyte, which is called microgametocyte, and the female gametocyte, that is the macro or the megagametocyte, that will be ingested. Even trophozoite stage, amoeboid stage, signet ring stage, these will also be ingested, but all these stages will get digested, and it is only the gametocytes which will be spared from digestion. Now the RBCs around them will be digested, and gametocytes are set free, in which there will be 2 types of gametocytes, as we mentioned, the female gametocyte and the male gametocyte. So these are usually alike. We have just shown them by different colors to distinguish them, but that is not easy to distinguish. Then next what happens is that the male gametocyte, it forms up to 8 male gametes by a process called exflagellation. So in this you see that the male gametocyte from this flagellar structures are coming out from the cell surface. There is evagination taking place, and in that the nucleus inside that gametocyte will divide and pass into each of that flagellar outgrowth, and all these flagella will then separate, so that 8 gametes are going to be formed. So that's why the process is called exflagellation. And where is all this occurring? It is occurring in the stomach or the midgut of the mosquito. Now next comes fertilization, which will be occurring here in the midgut, and a zygote is going to be formed. This zygote is the first diploid stage, because Plasmodium itself is haploid, where the 2 gametocytes had formed gametes. At that level there was no meiosis. So it was just the entire organism itself working as a reproductive unit. Now those 2 reproductive units have fused together to form a diploid zygote. And this zygote then acquires a worm-like motile shape, and it is called ookinete. Now ookinete is something very specific to this Plasmodium, a motile zygote being formed, and this ookinete, it bores into the wall of the gut, the midgut of the mosquito, and however it doesn't come out into the hemocoel, but rather remains in the external covering, or you can say just the subdermal region. So growth and division will be occurring in this ookinete. So it will first transform into spherical structure called oocyst. So it will have covering around it, a cyst wall around it, that oocyst, and this oocyst will undergo meiosis, so that again a haploid chromosome number is achieved. This will then undergo division, very similar to schizogony, but now this division is going to be called sporogony. And the daughter cells which are going to be formed inside this oocyst, they are called sporozoites. So this level of division after sexual reproduction, this cycle is sporogony, and These are present on the gut wall of the mosquito. These will be liberated by the rupture of this oocyst, and these will come out into the hemocoel. From the hemocoel, they will invade into the mosquito salivary gland. Now this mosquito has become infective. So such mosquito can then bite a human and cause infection. Now there is few more things we need to discuss about malaria. So we will be discussing regarding the treatment and how to control malaria. We have discussed the life cycle of Plasmodium vivax, which is one of the common malarial infection. But let us compare the other Plasmodium species as well which cause malaria. Now in this, taking up pre-patent period, now this is the schizogony going on in the liver, during which time there are no symptoms in the human body. So for vivax, this period is of 8 days, for falciparum it is 6 days, for ovale 9 days, and for malaria it is 13 days. Now the other one, that is erythrocytic phase, that is the time taken for the cycle to complete in the RBCs, that is 48 hours for vivax, falciparum as well as ovale, but for malaria it is 72 hours. Now coming to the incubation period, that is the day when sporozoites are injected into the body till the appearance of symptom, that is the first rupture of the RBCs. Now this time period is 14 days for vivax as well as ovale. For falciparum it is the least, that is 12 days, and for malaria it is the maximum, that is 30 days. The type of malaria which is caused by these, for vivax it is called the benign tertian malaria. Benign because it is not fatal, it doesn't affect the life of the person. And why tertian term we are using? Because the erythrocytic schizogony is completed in 48 hours, that is 2 days. And the next symptom, that is fever with chills, will appear on the 3rd day. So that's why we are using the term tertian. In case of falciparum, the malaria is called malignant tertian malaria or the cerebral malaria. For ovale, it is mild tertian malaria, and for malaria, it is quartan malaria, because there you see the erythrocytic cycle is of 72 hours. That means 3 days. So next symptom will appear every 4th day. So this way we can see a comparison between the various Plasmodium species. And also one more thing that we had mentioned, that is in falciparum the relapses do not occur, because once it has liberated from the liver, it will not return to the liver. So that's why there is no relapse in falciparum. Now moving on to the treatment part. This is one common treatment medicine, that is quinine, which is a very bitter alkaloid obtained from cinchona bark, and this cinchona tree is producing quinine, and it is effective in treating malaria, but only those phases of malaria which are in the blood. It doesn't have any effect on the liver stages. And chloroquine or chloroquine is the usual medicine of choice, but nowadays there are number of strains of this Plasmodium which are immune to quinine and they are not being controlled. And in recent times, a new medicine, that is artemisinin. So artemisinin based combination therapy or ACT, this is the current WHO recommended first line treatment for the majority of malaria cases, and this medicine is useful for all stages of the parasite and also the all species of the Plasmodium, even falciparum malaria. And this artemisinin was discovered by a Chinese scientist Yu Tu, for which she got the 2015 Nobel Prize. And also the control of the vector, that is the mosquitoes. For that, we have introduced a larvivorous bony fish in the ponds, and that fish is Gambusia. This fish exclusively feeds on the mosquito larvae, and that is why it is useful in controlling the mosquito population. So this was regarding malaria. Now moving on to another protozoan disease, that is amoebiasis or amoebic dysentery. Now this disease is caused by Entamoeba histolytica. Now this one is an amoebid, that means the protozoan which move by the help of pseudopodia. However, Entamoeba, since it's a parasite, so there is only one single pseudopodium on its cell. Now, amoebiasis is transmitted by contamination of drinking water and the food with the feces, but it can also be transmitted indirectly through contact with dirty handling of the food or the objects, and once again, houseflies can be the mechanical carriers for this disease. Now, the infective stage for the humans is quadrinucleate cyst. Now, when we say quadrinucleate or tetranucleate, that means within the cyst there are 4 nuclei, and along with those 4 nuclei there are 2 dark staining chromatoid bodies which are actually ribonucleoproteins. Now once these cysts are ingested along with food and water which is contaminated, there is excistation, that means the cyst wall will dissolve, and that will be by the action of trypsin in the small intestine. And from this cyst, a single amoeba emerges. Now, this is an important question, that where does excistation takes place? So, that is in the small intestine, and the number of amoeba here I am using as a common noun, that is entamoeba, and the number of amoeba which are emerging out is only 1. And this one can be called metacystic amoeba. Now, this metacystic amoeba, it moves to the lower region of the small intestine. From there it will be passing to the large intestine, and it divides by binary fission 3 times, one after the other. So there will be 8 small amoebulae formed, and these 8 small amoebulae can be also the trophozoite stage, that is the feeding stage. However, out of these trophozoites which move to the large intestine, some would be larger, which are called the magna form, and that is the infective stage, while some of them will be the minuta form, which are the non-infective or the non-pathogenic stage. Now this magna form will penetrate into the mucosa of the large intestine, that is the colon, and from the mucosa it can reach up till the submucosa. So it will be causing damage to the lining of the intestine, while this minuta form, it remains in the lumen of the intestine and it undergoes encystation. So it will form initially a uninucleate cyst, then a binucleate cyst, and finally a quadrinucleate cyst, which will be passed out along with the feces. Now this magna form, which has penetrated through the wall of the colon, it will be feeding on the tissues over there, and in the submucosa we know that there are blood vessels, those will also be ruptured, and it will be feeding on the RBCs also. So this will be causing ulcers over there. Now the invading trophozoites, they destroy this epithelial lining, and there will be ulcers characterized by minute points of entry, and then they are becoming bigger, so it will be almost a flask shaped structure formed by this penetrating magna form, or you can say it is like a crater formed by dropping a bomb. And the symptoms of amoebiasis are highly variable, ranging from asymptomatic infection where there are no symptoms at all to dysentery along with blood, and even there would be episodes of constipation. So sometimes there is dysentery with blood, and then sometimes there is constipation, and there is lot of mucus also along with the feces. Appendicitis can also occur, and abscesses, that is pus-filled spaces, can be created in the liver, lungs, or the brain if through the blood vessels in the submucosa the amoeba reaches to other parts of the body, and that is sometimes fatal also if it reaches the brain. Now, amoebiasis can be prevented by proper sanitation. If there is proper disposal of the waste matter, then certainly amoebiasis can be properly prevented, and that is happening in the developed nations, but again the developing nations have still to improve their sanitation. Now, from these protozoan diseases, we move on to the next category of diseases, and those are the helminth diseases. Now, you remember helminths or helminthology is a term for platyhelminthes, in which we have parasitic forms like tapeworms and the flukes, and the other one is nemathelminthes, in which we have parasitic roundworms. So here we are discussing some of the important helminth diseases. Many of these we have discussed with the animal kingdom. So one of them is Ascariasis, which is caused by Ascaris lumbricoides, which is a roundworm pathogenic to humans. The infective stage is the embryonated egg. Now this embryonated egg, if you see, this is having outer cyst, and within that cyst there is a juvenile of this Ascaris, and this juvenile is the second juvenile. So that's why we can say the second juvenile or the second rhabditiform larva, which is enclosed inside this cyst. So this one is the infective stage. If any other stage is ingested, that won't be infective for us. And how can it go into the body? That would be through contaminated food and water, and also fruits, vegetables if they are not washed properly, and sometimes when children, they are playing in the open grounds and they don't wash their hands, and these cysts may have got into their nails or hands, and those are used to feed themselves. Now the cyst will be digested in the small intestine, and the juvenile will be set free. Now this juvenile will migrate from the intestine to the liver, and from there to the heart, then the lungs, and finally it will be returning to the intestine. And during this migration, it will be undergoing various levels of molting, and it will transform into adults. So we can see the Ascaris adults, the female is larger, the male is shorter, and in this case the symptoms of Ascariasis will be, there is internal bleeding can occur, muscular pain, fever, anemia, and blockage of the intestinal passage. If there are too many worms in the small intestine, the passage of the chyme is affected. And these adult worms, they feed on the chyme. And the eggs of the parasite, they pass out in the feces of the infected person, and they will be contaminating the food and water of the other through the soil. Another helminth disease is filariasis. Now this one is caused by, again, roundworms, Wuchereria bancrofti, and another species is there, Wuchereria malayi. And these are transmitted through the bite of the female Culex mosquito. And in extreme cases, it can result in elephantiasis, where the parasite is lodged in the lymphatic system and the lymphatic return is no longer possible, especially in the parts which are on the lower side of the body, that is the lower limbs and also the genitals. Now this filarial worm is quite common in our country, and because the mosquito population is also high. So even this disease can be controlled if we are able to control the vector. So after taking up these helminth diseases, we'll have a discussion on one last category of diseases, that is the fungal diseases. next disease among the category of common human diseases are taken from the diseases caused by fungi. Now, one of the important fungal disease in humans is ringworm, or which is also called tinea. Now, the fungi species which cause ringworm are Microsporum, Trichophyton and Epidermophyton. Now, these fungi, they feed on keratin, a protein found in our skin. Now, this ringworm is highly contagious. So, one person who is infected, when they come in contact with the other, even that person can get infected by the disease. It can be passed from one person to the next by direct skin to skin contact or by contact with contaminated items such as combs, towels, clothes, and even the shower or the pool surfaces. Now, this disease, if we see the person who is affected, there would be a reddish colored scaly patch formed on the skin. So, whichever part of the skin is affected, there would be a reddish scaly patch, and that will have intense itching in it, and it will be flaky so that some dead tissue will be coming out of it. The patches are often redder around the boundary, and they are having a lesser intensity color in the center, and that's why it gives the shape of a ring, and that's how the disease is so named. And bald patches develop if a scalp or the beard is infected. So whichever part of the body is infected, accordingly that ringworm is named. Like it can be Tinea capitis. So capit term is for the head, so it is the scalp which is infected. Tinea corporis, that could be any place on the body. Then Tinea pedis, that is athlete's foot, which will be between the toes. And there are antifungal creams by which one can treat the disease. Now, after going through diseases, we can discuss a few points regarding how to prevent. So, what can be the various preventive measures against the communicable diseases? Now, one important is, of course, education. So, the people, they need to be educated about the communicable diseases so that they can protect themselves. The other is isolation. For communicable disease, the infected person has to be separated or segregated from the rest of the healthy people so that the disease can be contained and it is not able to spread. Vaccination. If for that disease a vaccine is available, then certainly timely vaccination will help to prevent the infection. Personal hygiene, keeping the body clean, and even that can help to prevent the further spread of the disease. Or if we are hygienic enough and whatever food and water we are taking, we are careful about it, then too the disease can be prevented. Along with personal hygiene, another important aspect is the public hygiene. That is the proper disposal of waste and the excreta, periodic cleaning and disinfection of the water sources, and that is also essential in preventing the diseases. Sanitation, that again is part of the hygiene. Garbage, heaps, polluted water, food exposed to the dust and the flies is the chief source of number of pathogens. So proper sanitation can prevent the spread of the diseases. And last but not the least, sterilization. So the patient's surrounding and the articles of use should be sterilized. So soap, phenyl, Dettol, etc. may be used whenever it is necessary. And even for water and food, if it is properly boiled at high temperature, then too the pathogens which are present in it will be killed. So this was one important part of this chapter. So we can conclude today's class with few of the questions. So let us take some questions. Our first question is, different species of Mycobacterium cause syphilis and diphtheria, whooping cough and leprosy, tuberculosis and leprosy, and lastly syphilis and gonorrhea. Now, Mycobacterium, we have studied two of these species, that is tuberculosis and leprae. So that means here the answer would be tuberculosis and leprosy. So this makes option 3 as the right answer. Now next question. A disease contracted through wounds, accidents and improperly sterilized surgical instruments is. Now in this case, the disease which is spread in this manner, that is tetanus. The other diseases we have studied, gonorrhea is through sexual contact, mumps is through droplet infection, amoebiasis is through contaminated food and water. It is tetanus which is spread through open wounds or by using instruments which are not properly sterilized. So this makes option 1 as the correct answer. Now moving ahead to the next question. The active form of Entamoeba histolytica, active form as we had seen the magna form, this one secretes a toxin also, histolysin, which breaks down the tissues and it feeds upon blood only, erythrocytes, mucosa and submucosa of colon, mucosa and submucosa of colon only, food in the intestine. So in this case, the correct answer would be erythrocyte, mucosa and submucosa of the colon. And we have mentioned that it penetrates through the lining of the colon and it feeds also on the blood cells. So here option 2 is the correct answer. Now moving ahead to the next question. Motile zygote of Plasmodium occurs in. Now the motile zygote of Plasmodium is called ookinete and the zygote was formed within the body of the female Anopheles. So where is it formed? That we can see over here. Now two options we can easily rule out, human liver and human RBCs. The other two options, gut of the female Anopheles and salivary glands of the Anopheles. Now salivary glands would be harboring the sporozoites, but the ookinete is in the gut and that too in the midgut, which is also called stomach. So here the correct answer is option 2. Next one. Where will you look for the sporozoites of the malarial parasite? Salivary glands of freshly molted female Anopheles mosquito. Now, mosquito has a complete metamorphosis in which the eggs which are laid in water will form a larval stage, which is the wriggler, and this wriggler will then undergo pupation, which is called the tumbler, and from that tumbler the adult mosquito will hatch out. Now as soon as it has hatched out from the pupal stage, it has not yet taken the blood of any malarial patient. So certainly it cannot be carrying the sporozoites. Saliva of infected female Anopheles mosquito, red blood corpuscles of humans suffering from malaria, spleen of the infected humans. So here the answer would be the saliva of infected female Anopheles mosquito. So here the answer is option 2. And as far as spleen is mentioned, spleen is the graveyard of RBCs. So during malaria, when RBCs are being ruptured, those dead RBCs are stored in the spleen. That results in splenomegaly. The spleen enlarges in size and the person also suffers from anemia. Now moving ahead with the next one. A person suffering from a disease caused by Plasmodium experiences recurring chill and fever at the time when the sporozoites released from RBCs are being rapidly killed and broken down inside the spleen. The trophozoites reach maximum growth and give out certain toxins. The parasite, after its rapid multiplication inside the RBCs, ruptures them, releasing the stage to enter fresh RBCs. The microgametocytes and megagametocytes are being destroyed by the WBCs. So in this case, we know that when RBCs rupture, hemozoin is released, and this would be the third one here, which explains the reason for recurring fever with chills. That is, the parasite, after its rapid multiplication inside the RBCs, has ruptured them and merozoites are released along with hemozoin. So the correct answer would be option 3. So this brings us to the end of today's class, in which we have studied some of the common human diseases caused by bacteria, helminth, protozoans and fungi. So this completes one part of the chapter. In the next video, we'll be taking up immunity, and we'll be discussing the various concepts behind immunity, that is the non-specific as well as the specific immunities. So thanks for watching. See you in the next video.